series of lectures that we've done as part of the RiziCulture rice research program here, a year-long interdisciplinary look at rice from a variety of different perspectives. Our faculty research group includes people from art through biology by way of history and demography and agriculture and whatever. So we've got a broad range of people looking at rice as a research question. And part of it, or a facet of that, is that we've been bringing in speakers from outside. We've had a speaker earlier this fall, Peter Corklanis, who came to talk about the history of rice in Arkansas and Louisiana. And today we had our second speaker who's come to talk to us about the rice breeding and its future and the potential for the use of wild varieties of rice to improve existing and future varieties of rice for cultivation. Before we kind of launch into that, I wanted to thank, we have a bunch of people or groups that have sponsored this program, and those include the College of Humanities and Social Sciences, the College of Agriculture, the College of Communication, which has provided videography for this, and we expect very soon to have the first lecture available, Peter Corklanis' lecture, available on a website. So if you missed that one and you're curious about it, you can go back and watch that. And we hope to have Dr. McClung's talk also up on the website pretty soon. And the College of Communications has been helping us with that. Nursing, business have also contributed to this, and Rice on Foods has made a donation to support the lecture series. And the Office of Research and Technology Transfer has helped support the faculty research aspect of it. So there are a number of different bits of the university that have contributed to make all of this possible and to make it happen. Tonight's speaker, Dr. McClung, comes to us from the USDA ARS Rice Research, the Dale Bumper National Rice Research Center in Stuttgart, Arkansas. She has been there since 1991.
With ARS,
in Texas, working at the Rice Research, USDA Rice Research Center there. She has her PhD from North Dakota State, and undergraduate degrees from Texas A&M, and a master's degree from Texas A&M. She's a plant breeder she worked after she got a PhD. She worked in private industry for a while, and then in '91 came to the USDA. She's produced 15 different cultivars of rice, and if you are familiar with various rice varieties, some of the better known of those are Presidio, Sabine, and Dixie Belle. And I was at a conference this spring, and one of the things I heard in one of the lectures there, or one of the talks there, was that Presidio is one of the 3 most widely planted varieties in the U.S. And so that's, it's a top 3 rice variety. Currently, the USDA Research Center in Stuttgart is focused upon market development for rice and for doing genomic work on rice with the intention of creating improved varieties, bringing in new types of genetic material to meet sort of new types of challenges that are potentially emerging in the 21st century for rice growing. So I will, at this point, turn things over to Dr. McClung, and she can take over with her talk. So, thank you. Now let's turn this off. So,
thank you.
Sure. I'll get you some water.
And is the mouse up here?
I'm not sure. I think you have to tab it with your.
Oh, this is the mouse, and then I meant the clicker. Oh, here.
Yeah. Now whether that clicker is connected to this, that's got the pointer.
I got so many things. Well, thank you very much for inviting me, Eric, and thank you folks for sharing your evening with us tonight. And it certainly is a pleasure to be here. I've visited the campus. I think this is my 4th time, and find new people, new departments, new opportunities each time. So I'm very pleased to have the opportunity to come, and excited about the research that we already have ongoing with some of the staff here, and looking forward to opportunities that we have in the future. So I'm very pleased to be in the lineup of some of the folks that Eric has said that he's going to have coming to the university to talk on rice from all different aspects, and hope that bringing you the perspective of some applied breeding and how we're trying to utilize genetic resources to kind of tackle some of the problems that we see coming towards us in the future. So Eric and I kind of came up with this idea, the topic here. So it's kind of long and wordy, but we're going to be covering a lot of ground this evening, going all the way from wild species to cultivated rice, and how those are being used currently by our farmers, and how we're going to be utilizing these resources in the future for our farmers. So let's see if I can do this. So what I'd like to do is just give you, first of all, an overview of rice production, and I know the university here is sitting in the heart of rice production in the northeast corner of the state. We'll talk a little bit about rice production, not only at a global perspective, but also bringing it right down here to our own state, and talk about some of the production challenges that we do have facing us, and then how we are preparing to utilize genetic resources to address these challenges and to improve food security into the future. So first of all, just a little bit of background about how important rice is in the world. This is showing these are the 3 top cereal grains in terms of their the area of where they're produced in the world, and you can see that rice and corn are currently neck and neck in terms of the planted area. And even though most corn is hybrid corn, you can see that the yield potential of rice is really quite high as compared to corn. And it's higher than wheat, but a lot of that is because, of course, wheat is grown in much wider acreage across the world and maybe under some environments that are not as conducive to high production. Whereas most rice is produced under flooded conditions, which helps to stabilize yields and increase yields. So that's one of the reasons why it has the higher yield potential. So in addition to it being grown widely around the world, it of course is a staple food crop for about half the world's population. And not only that, this chart shows that these are the countries that have a high proportion of their total calorie supply is based upon rice. And so you can see that there's some countries like Cambodia, Bangladesh, Myanmar that have over 70% of their total calorie intake is based upon rice. And so obviously if we have situations where the price of rice changes dramatically, which the people that are going to be impacted most dramatically are the folks that live in some of these third world countries and are so highly dependent on rice. So global food security, and as it relates to rice, is critically important really for stability into the future for our world. So within the United States, even though we don't think about it as having a large acreage in terms of production, it actually has it's a very significant player in terms of the amount of rice that's exported and available for world trade. The biggest exporters are Vietnam, and then Thailand, and this is primarily what they call jasmine rice, which is an aromatic. And then India is another huge exporter, and that's largely the basmati style rice. And then the United States, because we are able to export about half of our production here in the United States, we're contributing about 10% of the rice that's in world trade. So even though we're small compared to some of these other major countries, we're in the top 5 in terms of exporters. So this is just looking at some of the acreage of rice and how that's changed over time. and the yield of rice, of how that's changed over the last 50 years or so. And you can see that yields have continued to increase, and that's for a variety of reasons: technology, better varieties, knowing how to produce the rice in the most productive ways. But we can also see that there's not an increasing amount of land that's being turned to rice. It's actually declining. And so we're getting more and more of our yield off of fewer acres. Okay. So this is from a recent publication that is looking at how rice yields are changing across the world, and over a period of, I forget how many years, but a number of years. And so this is kind of a heat map, and it's showing that areas that are colored in red or yellow are where they're actually showing decreasing yields, whereas the areas in green are stable or improving. And so you can see that the southern United States is green, and it's actually considered a high productive areas. But some of these other areas that are so critically dependent upon rice consumption are actually experiencing declining yields. And so as populations increase, this is going to be a very critical problem that has to be addressed. So we are seeing changes over time in terms of rice production in the United States. Rice is grown primarily in the southern Mid-South, which is Arkansas, Louisiana, Mississippi, Missouri, and Texas. And also we have an area in California. And so this shows essentially from the beginning of rice production in the United States how the acreage has changed over these 100 or more than 100 years. And so here we have blue. Arkansas has increased, but it has there's been a high fluctuation, and of course the amount of acreage that's put into any crop is dependent upon a number of different reasons, economic reasons, marketing, alternative crops that can be grown in that year, and the price of the crop. But what I wanted to show is that particularly if you look at Texas, it has had a major decline in acreage. Its highest acreage was 540,000 back in 1954, and so it's now well below 200,000 at this time. And likewise, with Louisiana, there's been some decrease in it, and where we've seen an increase in acreage in terms of the Mississippi area and Missouri over this period of time. So as you look at what's, the type of area on the Gulf Coast where rice was originally grown, there's a lot of industrialization and a lot of urbanization. And so what we're seeing is, to some extent, that's pushing and competing for that land. And so rice is moving into other areas as one of the drivers. So what are some of the aspects that are actually impacting rice production and rice yields? And as we look at these factors, if we want to sustain rice production in our country or in any country, of course, we have to sustain rice farmers. And farmers are in it to make money. This is their livelihood. And so I've just listed here, certainly not all of the factors that are affecting essentially inputs or resources that go into production. So we have having access to productive land, their ability to control various pests, having new rice varieties that will have high yield and good quality, having access to water, both in terms of quantity and good quality of water. Of course, fuel prices have a very dramatic effect on so many of these other aspects. Having access to labor and to new equipment, and also having access to fertilizers to maintain these high yields. And I recently was at a presentation where it was said that 90% of the potassium, which is the K that is used in fertilizer, is imported to this country. And so as we see and one of the primary sources of that potassium is Russia. So as we see other parts of the world begin to maybe increase their productive capabilities and their mechanization and trying to increase the yields of their crops, we may be seeing increasing competition for access to these critical nutrients for production. And so on the profit side, of course, some of the factors that are going to determine that is the crop value, which will be for rice, both the yield and the milling quality that comes from that. And of course, their ability, a farmer's ability to compete in both global and domestic markets. And as I mentioned, right now, about half of our production is exported. It used to be that the United States was considered the premier quality rice in the world. And whenever our rice was put out on the market, it actually received a premium price. However, what we've seen over the last 20 or so years is that these other countries have made dramatic improvements in their quality. They're shifting from not just improving yield, but also trying to improve their competitive advantage in the market. So this is getting more and more difficult for our farmers to compete on the open market, and certainly is something that we're going to have to put more and more emphasis on. So these are the areas within the country where we have the rice production. And as you folks all know, that Arkansas produces about 45% of the country's rice. And I've identified here some of the major issues that are kind of recently have been affecting rice production. Here in the Arkansas, one of the biggest issues that our farmers are facing is access to water. Essentially all of the rice in the Arkansas area is being irrigated through pumping from underground aquifers, and many of these are being depleted. So having access to water for rice production is certainly a major issue to the future. In the Louisiana area, which produces about 16% of the nation's rice, there have been, in just recent years, some 20,000 acres that have gone out of production right along the Gulf Coast as a result of saltwater intrusion. And some of this is due to just the loss of the marshlands, which is a protective barrier for saltwater intrusion, as well as when we've had some hurricanes recent past that have pushed salt water up into these areas. And once the fields become inundated with salt water, they may be out for a number of years. This past, in 2011, there was a huge drought in Texas, the rice growing region. And essentially in Texas, rice is produced on either side of Houston. And this western area, what they call it west of Houston, some 50,000 acres were not even planted to rice because there was just not access to water. So there's frequently issues in California. Their irrigation is dependent upon winter precipitation, snow in the mountains. And so these are some things that are definitely affecting acreage in our country. And as we look at issues like associated with climate change, we can see that these are going to become maybe more dramatic problems in the future. So I mentioned that within Arkansas, there's a problem with access to water from the aquifers. And so what this is a map here showing rice production, the states with the counties in Arkansas with the most rice production. And so you can see here that we have Lawrence, to Jackson, Cross, and Arkansas. Those are the highest, some of the highest producing areas, as well as Poinsett County in terms of their ranking of rice production. Here's a map of Arkansas, a heat map essentially showing the depth of the aquifers in these areas. And so the red colors means that the depth to reaching the water underground is deeper than where it's the blue color. And so these areas that are considered, that are red, they're called critical groundwater areas. And you can see that they overlay essentially completely on the counties where we've had the highest rice production. And so rice is one of the biggest users of these underground water aquifers. And this is just not a sustainable system for rice production into the future. These aquifers are not being recharged as fast as they're being depleted. So this is probably going to be one of the biggest issues facing continued rice production in Arkansas into the future. So one of the ways that they're approaching this is they're essentially trying to help farmers get to the point where they're switching from using underground pumping of water to using surface water. And so part of that includes major water diversion projects, diverting the White River throughout the rice growing region through canals, building of canals and using existing canals. And also a number of farms are putting in large water impoundment areas on the farm. So essentially if rain falls on your farm, make sure that that rainwater doesn't leave it. That's that water that you can use for irrigating your farm. So if we continue to produce rice in the way that we have in terms of a season-long flooded system, you can see where access to water is probably going to be a very critical issue for farmers into the future. So the way this is done through our conventional flooding is essentially the field is planted, and then as the rice begins to emerge, that the field becomes flooded, and as the rice gets taller, the flood continues to get a little bit higher to the point where it's being used essentially to control weeds in the fields. However, there's been some work that research that has been done worldwide and is starting to be done here in the southern U.S., where they're using, and I know many of you have seen these poly pipe that's gone through the field, and this is what they call multiple inlet. And so this allows faster movement of water into a field, so rather than flooding in from one place and moving it all the way through all the different fields across the farm, this allows faster movement and flooding of the field. And so there's been some research that's been done primarily in Mississippi, where they're actually using what they call intermittent flood, or also known as alternate wetting and drying. And so you're flooding the field and leave the flood on there and then allowing it to evaporate to the point where the ground is actually dry, and then they'll flood it again. So rather than putting the extra resources to maintain the flood throughout the year, it goes through this intermittent flood process. So of course, one of the concerns for farmers would be what would be the impact upon yield and milling quality. And so the researchers in Mississippi have shown that they have been able to maintain the yields very similar to a flooded system, but having significant reduction in the amount of water that's being used. So this may be something that needs to be looked at a broader range of environments and soil types in the southern U.S. to save water resources. So in addition to having the challenge of having access to water, another concern is about temperature change. And so this is looking at temperature from 1975 and projected to 2015 for the Louisiana and Mississippi area. And here we have the number of days that are less than 32 degrees Fahrenheit. And here is the growing season length, and at the top here is the yearly minimum temperature. And so what we see is that the minimum temperature, which is generally considered like the nighttime temperature, over this period of time has been increasing, that the growing season has been lengthening. We have longer seasons. And the number of days that we have that are cold days are decreasing. So the impact that these changes, these trends in temperature change that we see is having in terms of on the rice crop is that we know that higher nighttime temperatures are going to reduce grain quality. And this is in terms of reducing the milling quality and increasing the chalk that is in the grain. The longer growing season, the way farmers are adapting to that, that allows them to plant earlier in the season. But what happens when you do that, there can be times when you have a lot more variability in terms of temperatures in the spring. And so sometimes those plantings may be subjected to actually cold temperatures as the crop is emerging and getting established. Of course, having milder winters will also result in maybe greater pest pressure in terms of diseases and some insects that would usually be knocked back during a colder winter. And having a higher frequency of hot days, of course, is going to put more pressure in terms of irrigation needs. And another concern is that having high temperatures around the flowering time of the crop may actually result in sterility and, of course, reduced grain yields. So that's some of the temperatures that have experienced already. This is looking at, again, some projections for changes in temperatures up to the year of 2100. And so essentially what we see here is that there's going to be more and more days that are projected to have that the minimum temperature is going to be higher. So more days are going to be higher. And you can see this is falling right along in terms of the areas where rice is very important in the southern growing region. So temperature impact is going to be another thing that we're going to have to deal with. In addition to that, rice, because it is grown under a flooded field system, it results in anaerobic conditions that allows bacteria in the soil to produce methane. And the rice plants essentially serve as like, the stems of the plants are like little straws that are pumping the methane that's down in the soil up through the straw and out into the air. So wherever their rice is produced under a flooded situation, there is the problem of methane emissions. And so there has been a number of discussions over the last few years about trying to reduce our carbon footprints. And so as we look at different crops and their carbon footprint and our nation's effort in trying to reduce greenhouse gas emissions, what we find is that with rice, it has one of the highest footprints for carbon. And so here, this is just looking at our current production practices and the estimated levels of greenhouse gas emissions. And again, as you'd expect, that the counties where we have the highest rice production are also the ones that have the highest greenhouse gas emissions. So in addition to the factors that I talked about that affect a farmer's ability to be economically profitable, we also have these big issues of climate change and policies by our government and other governments that may affect market prices and trade opportunities. So all of this are things that we are going to have to be dealing with in the future. So this note here is to remind me to tell you, don't leave the room yet, because it's not going to be that depressing. That these are just the challenges that we have facing us, and that we're going to need bright people like you that are in the audience to help us deal with these challenges. So this looks like a job for science to the rescue. And so what I want to turn now to is some of the things that we're doing to try to address these challenges that we have. So at the Del Bumpers National Rice Research Center, our research is completely focused on rice. And one of our main objectives is really to look at natural genetic diversity that exists in rice and try to utilize that to address whatever production issues or marketing issues may be important to the U.S. rice industry and the U.S. rice consumer. And so as we talked about these previous issues, what we need to be looking at is developing crops that are resilient to fluctuations in climate, and that are crops that can be adaptive to changing production systems. So first of all, just to get a little bit of background about rice itself, Oryza sativa is the genus and species that is the rice that is cultivated most throughout the world. But there are 23 other different rice species, or Oryza species. And these come from different, what we call, they have different genomes. This is the diploid AA genome, and then we have some other genomes here. And so what I'm going to be talking about is really focusing on the AA genome, and that's because these species, because they have the same genome as the cultivated rice, can actually be crossed easily with Oryza sativa and be utilized in terms of genetic improvement. So first of all, I want to talk a little bit about the diversity that we have available to us in Oryza sativa. So the USDA maintains a collection of some 18,000 different rice cultivars here in the United States that have been collected from all over the world. And of course, rice is not indigenous to the United States, and so you can see that we've been able to get rice from where it originated in the Southeast Asia area, as well as from all these other continents. And so we have this tremendous resource of diversity, genetic diversity, essentially in a gene bank here in the United States. And these entries go back to, some of them were deposited over 100 years ago. So not only do we have diversity across land, but we have diversity that goes across time. So much of this diversity really has not been utilized in terms of breeding programs, and that's because the U.S. rice breeders have worked very hard for a century to get rid of a lot of junk and really come up with some really good rice varieties. And so when they look at a field like this, which is an example of some of the diversity that's in the collection, they go, there's a lot of stuff out there I'm not interested in. And so one of our tasks is to essentially characterize this genetic diversity, both for traits, and we're now in the process of characterizing it genetically and identifying the genes that control those traits to make that material more useful to breeders. So one of the ways we do this is, evaluating 18,000 of anything is difficult. And so what we do is that we have developed what's called a core collection. And so out of these 18,000 varieties, a subset has been pulled that are representative of the countries that are in the collection and are represented from the varieties that have been offered across different time points. So both recent introduction as well as very old ones. And so we have a core collection that is about 10% of the whole collection, about 1,700 lines. And out of that, we've also developed what we call our mini core collection, which is only about 200 lines. And so when I come to some of the researchers here at ASU and talk about, let's do some genetic evaluation, nobody wants to talk about, if you're talking about 18,000 rice varieties, but if you're talking about 200, then they're more interested maybe pursuing something. And so what we've been able to determine that these subset collections actually represent 88% of the diversity that's in this whole collection. So these are reasonable subsets that really capture the genes that are available in the whole collection. So one of the things we do is just evaluate that collection for various traits. And here's a study where we looked at the collection for its ability to germinate under cold temperatures. We looked at some 2,300 different rice varieties, and as I mentioned before, that if we have farmers that are growing rice, planting rice earlier in the season, having rice varieties that are very vigorous under cool temperatures is important. And so here's a number of varieties that had very high germination, even at 12 degrees C. And what's kind of interesting here is that we've been able to identify rice varieties from very tropical areas as well as very more temperate areas that have this ability to germinate under cold temperatures. So this just shows that you can't necessarily go to a particular region or country for looking for a particular trait, that those genes are intermixed throughout the collection, coming from all over the world. So our next venture is not only to be looking at the phenotypic traits of the collection, but then of course understanding the genetic content of these cultivars and how to relate that genetic variability with the traits that are ultimately expressed in the plant. So just very briefly, what we try to do is that we both evaluate the material in the field for traits or under various laboratory conditions. We will extract DNA from the varieties that we're interested in looking at. And then that DNA is essentially chopped up into little pieces, and we're able to analyze that DNA and be able to determine the genetic variability amongst all of these different rice varieties. And so just as an example, this would be, for example, one gene, and it's from 2 different rice varieties, and this just shows that there's a difference between these 2 rice varieties in terms of the marker allele associated with that gene. And so what we try to do is when we're collecting this type of information over hundreds, if not thousands of genes, and then we're trying to relate that variability with the variability we see in the traits. So just simplistically, if this were a trait that was a gene that was controlling late maturity, like we see here in the field, that maybe this allele would be associated with the late maturity, and this would be another marker allele that would be related to early maturing. And so this would allow us to have a marker once we found that it was connected or correlated to the actual phenotypic trait, that we could actually use the marker in terms of breeding selection, then actually going out in the field and evaluating the trait. Now, of course, the objective is not so we can just stay in the cool air conditioning, but it's so that we can actually be more efficient and effective in our evaluations, doing this based upon the genotype versus the phenotype. And so what we're essentially doing for all these different varieties, we're developing a DNA fingerprint. And so I kind of try to think about if this is the chromosome and we have different genes along that chromosome, these are somewhat like a train with different cars on that train, and each one of these train cars may actually be related to controlling heat tolerance, disease resistance, or cooking quality. And so what we're trying to do is identify genetic markers that are linked to each of these different train cars, and by monitoring the markers we can actually combine different ones of these train cars into coming up with a new superliner train that would be a new variety. So by having that sort of genetic information, we're able to scan essentially hundreds of these different rice varieties. And what this has allowed us to see is that RiziCulture actually can be subdivided into 5 subpopulations. The temperate japonicas, which are what you found mainly in the temperate regions like in Japan, the tropical japonicas, and this is essentially where all of the US rice varieties come from. And then there's a group called the aromatics, like the basmatis. And then we have the indica group, which comes from Asia, and another group called Aus, which is a different season of growing season in which the indicas grow from. And so what this shows us is that within RiziCulture there's tremendous genetic diversity. And what breeders have done, they've tended to kind of work within their own little gene pool because they don't want to have, they essentially think that or have seen that some of these other gene pools have traits that they're not interested in. So what we're trying to do is with this genetic information is actually utilize those markers to help us make maybe wider crosses across the gene pool, or even wide crosses within the gene pool that would help us to maximize genetic diversity in our breeding. And so this is just kind of an example of what we're able to do now. These are 120 different varieties, these different rows, and this is essentially looking across one chromosome in rice. And so this is the complete genome sequence, you might say, across that area of the, that chromosome. And so the different colors represent different genetic backgrounds. And so by having this genetic information, we're actually able to put these rice varieties into these different gene pools. And so I know it's a little bit hard to see, but I'm sure you're familiar with what's called the semi-dwarfing gene, which was used to reduce the height. It was used both in wheat. It was the basis of the Green Revolution in wheat and also the Green Revolution in rice. And so what we see here, this is where that gene is located. It's on chromosome 1. And in the tropical japonica material, which are the greens here, that introgression of that gene, the semi-dwarfing gene, came from an indica species. And so it shows up very clearly where that is located in the genome. So this is a way that we can not only identify genes, but also identify what was the source of material where those genes came from. So we can do that not only on one chromosome, but all 12 chromosomes. And here's looking at those same subpopulations. In addition, what we have here is, in addition looking to the Oryza sativus, we also here are looking the Oryza rufipogon, which is a wild-related species. And the genetic material in this species is completely different than what we have in sativa. So this gives us new opportunities of new genetic diversity to possibly utilize. And just as an example, here's a variety T-tep from Vietnam that has been utilized. It has an excellent source of blast resistance, and that was identified back in 1978. And that very same gene for resistance that is in T-tep is now in a number of rice cultivars here in the United States that have very strong resistance to this disease. So I've been talking about Oryza sativa and a little bit of the rufipogon, but many of you have probably seen rice fields that look like this. And so what this is red rice, which is a weedy species, and it's actually sativa also that occurs in the field. And this is a weed that is essentially impossible to kill because it's so similar to regular cultivated rice, there's no herbicide that's selective for it. So until we had what's called herbicide-resistant rice, this was very difficult for farmers to get rid of this weed. And the problem is that it has this red bran on it, so when they send the rice to the mill to have it milled, it's a mixture of both the red bran and the white milled rice, and so that actually decreases the value of the crop. So even though this has been a problem, we're also finding that this weed that we've been trying to get rid of actually may have some traits in it that are desirable. And so this is some work that's just been accepted for publication from Katie Lisko and Dr. Lorenz's lab here. And so what they have done is looked at a small set of different rice varieties for differences in vitamin C content at these different growth stages. And so what this shows that among this little set of rice varieties that there is quite a bit of variation in vitamin C content. And the reason why that's interesting is because the vitamin C has been associated with tolerance to stress, different kinds of physiological stresses. Another thing that's very interesting is that here at this growth stage of the plant, which is kind of near after flowering, that the two lines that had the highest level of vitamin C were actually two weedy red rices. So this suggests that the weed that we're trying to get rid of may actually have some genes in it that would be useful in stress tolerance in breeding. This is another study that we've done with a scientist in Beltsville, Maryland. It's essentially showing the same thing. We have a group of weedy red rices compared to a group of rice cultivars, and they were grown in growth chambers under low CO2 levels and under high CO2 levels. And what this shows is how rapidly or how how quickly the plants grow. And what this shows is that the weedy rices under higher CO2 levels actually outgrow the cultivated rices. So this says that if we're in a situation under climate change where we're having higher CO2 levels, higher greenhouse gases, then it suggests that the weeds are actually going to be responsive to that and grow faster than the cultivated rice. So the bad news is that might suggest that the weeds are going to be more difficult to control. The good news is, well, gee, if we could capture those traits and move those into our cultivars, we may have more resilient rice cultivars. I'll just skip that, it's about the same thing. So here's another thing where we looked at in terms of, as I mentioned, that growing rice under flooded fields, there's methane emissions from that sort of production system. In this past 2 years, we've looked at genetic diversity for methane emissions under different rice varieties. And so here we've got a group of rice varieties looking at their season-long methane emissions, so these colored lines here. And what we found is essentially two groups. We have a group of lines that have pretty high emissions levels versus a group of cultivars here that have very low levels of methane emissions. We don't know why they differ, but certainly what we'd like to do, of course, would be able to identify the genes that control that and put that into new rice varieties. So in addition to looking at a Rhiza sativa species, there's these other wild species of rice that we're also trying to explore for utilizing their genetic diversity. And so these are some of these other sativas here that I'm going to be mentioning that are also in the A genome. And again, these are ugly looking plants, only their mother could love them. They have no redeeming qualities as far as agronomic characteristics, but what Dr. Georgia Eisman going to do is she is crossing these varieties with some of our existing rice cultivars grown here in the United States. And the reason why we're interested in doing that is previous work has shown that these wild species of rice may harbor some resistance to various insects and diseases. And so we want to see if we can transfer some of those traits into cultivated rice. And so this is just a survey of some of the rice from the Rhiza rufipogon. And so this shows that there's just as much genetic diversity among this different biotypes of this one species as we saw in the actual Rhiza sativa. So tremendous amount of genetic diversity. And this is an example that when you cross a rufipogon, which is the wild species, with the Rhiza sativa, you get an F1 hybrid that shows tremendous amount of hybrid vigor. And that just says that there's a lot of genetic differences between these two species. So I've been involved with a project with Cornell University with Susan McClung, and one of our questions was, in addition to pest resistance and disease resistance, could these rufipogon, could it actually be a source of yield enhancing genes? And so what we did is we had a rufipogon donor parent that was crossed to the rice cultivar Jefferson, and we developed what's called a mapping population. And what we found was that there were a number of chromosomal regions in rice in this mapping population between these two species that were associated with yield enhancing attributes. And so what we wanted to do was then to go through what's called a backcrossing program to kind of isolate these different genomic regions and truly identify what their individual impact was. Oops. Not sure how to go back. Oh, went that way. So this is just showing where this is an example of the white would be kind of the Jefferson background, and the black would be the introgression from the rufipogon. And so looking across the different 12 chromosomes, we have these families that have essentially a Jefferson background, but just a few pieces of the rufipogon. And then we went through a yield test. And what we found is some of these introgressions on chromosome 2, 3, and 6, and 8, and 9, that we had 17 to 20% yield increase compared to the Jefferson parent. So this is just showing again that this weed, when you cross it in there, it actually has some yield enhancing traits also. So in summary, we have a number of challenges that we're having to face in terms of what our farmers are going to be facing in the future. This includes temperature extremes, limited water, and reduced water quality. And so this actually may be the bottleneck to the livelihood of our rice industry in the future. And so these are the challenges that we're going to have to deal with. However, we have a tremendous amount of genetic diversity that can help us address these challenges. And we now also have some of the genomic tools that are going to make using these species, which in the past would be just almost impossible to get any good value out of them. These tools are actually going to really help us expedite using those materials. And so these collections and genomic tools are truly very valuable. But what we're seeing now is that the genomic or the genetic sequencing that's taking place has now gotten to the point where it's so inexpensive that there's just this tremendous demand to have phenotypic information, good quality phenotypic information to relate to that. So that's kind of right now is where the real push is in our area, is trying to get more and more good trait data that we can help us identify and utilize these genes. I'll close with just identifying my collaborators: Susan McCouch at Cornell University, Lou Ziska, who's with ARS in Beltsville, Maryland, Georgia Eisenga and Dave Gailey, who are with me at the Dell Bumper Center in Stuttgart, and Dr. Lawrence, who's here at Arkansas State University, for their contribution to this work. So thank you. I appreciate your time this evening.